Greetings everyone and welcome to another Genetic Engineering and Biotechnology News webinar. Our presentation today is entitled Decoding Transcriptional Regulation by Genome-Wide Reporter Assays and Large-Scale Transfection. I'm Jeff Pogaliskas, Technical Editor for Gen, and I'll be the moderator for today's webinar presentation. As genomic technology continues to advance, the knowledge surrounding gene regulation is becoming less enigmatic. Yet the identification of active regulatory elements remains challenging, particularly for distal enhancers. Traditional methods for directly measuring enhancer activity are limited by throughput, so conventional approaches largely depend upon indirect methods that highlight features correlated with regulatory elements. Let's meet our speakers for this webinar, who will describe two novel ectopic plasmid-based massively parallel reporter assays that can directly and quantitatively measure the activity of millions of candidate sequences for enhancers and core promoters. Additionally, our presenters will tell us how these reporter assays were enabled by MaxCyte's scalable flow electroporation technology to attain high transfection efficiencies of plasmid reporter libraries. Our first presentation today will be given by Pierre Heine, a European field application scientist with MaxCyte. Dr. Heine will provide a brief introduction to MaxCyte's flow electroporation technology and its advantages in scaled-up transfection methods. Our second presentation will be given by Vanya Cosmos, postdoctoral researcher at the Research Institute of Molecular Pathology in Vienna, and Cosmos Arnold, a research associate also at the Research Institute of Molecular Pathology. Drs. Haberl and Arnold will describe the advantages of their novel reporter assay called STARR-seq and STAP-seq and how they can be used for the quantitative determinations of enhancer and promoter activities. Before we begin with Pierre's presentation, I want to encourage everyone to submit questions for our Q&A session at the end of the talks. We'll try to get to as many questions as we can, so simply type your question into the Q&A box on the left-hand side of your screen and hit submit. Okay, with all the particulars out of the way, let's get our webinar started. Pierre, the audience is listening.
Hello, I'm Pierre Heine with MaxCyte. I would like to start by welcoming all of you to our webinar. Before introducing our main speakers, I'd like to spend a few minutes to tell you about MaxCyte and the unique approach that we have taken to cellular transfection. Our technology was developed for clinical use, and therefore our focus has always been on providing a safe, non-viral approach to loading molecules into challenging cell types with high efficiency while maintaining high cell viability. MaxCyte's cell loading technology is based on electroporation. We suspend the cells in a physiologically balanced conductive buffer with relevant loading agents, and then apply a series of electrical pulses that create a temporary perturbation to the cell membrane, allowing molecules of almost any size to enter the cell very rapidly. Our motto is any molecule, any cell, any scale. In addition to plasmid DNA, we can efficiently load mRNA, siRNA, and even proteins into many different cell types. The technology can be used with a wide range of primary cells and cell lines, including insect cells. One of our technology's most unique features is scalability. We can transfect small numbers of cells, 500,000 cells up to a few 100,000,000 cells by static electroporation, which takes only a few seconds. Larger numbers of cells between 500 million and 200 billion cells can be transfected by flowing cells through the electroporation chamber. Because we apply the same series of electrical pulses for every transfection with a given cell types, we achieve very consistent results regardless of the scale. The MaxCyte delivery platform has been adopted by many pharmaceutical companies, biotechnology companies, academic institutions, and other organizations to develop a range of therapeutic modalities. We can generate modified cells for autologous and allogenic cell therapies. We enable large-scale manufacturing of viral vectors and VLPs for vaccine and gene therapy applications. We can produce high-titer antibodies and other protein-based biologics in cell types that are relevant for bioproduction. And we can express complex drug targets, such as ion channels and GPCRs, in large numbers of cells for cell-based drug screening. MaxCyte offers two different instruments for basic research, bioproduction and high-throughput screening applications. The MaxCyte STX instrument can transfect as few as 500,000 cells or up to 2 × 10⁻⁻⁻ cells at one time. Smaller cell numbers up to 700 million cells are transfected in a few seconds. Larger cell numbers flow through the electroporation chamber. During electroporation, cells are suspended in MaxCyte's chemically defined electroporation buffer at high density, typically 100 and 200 million cells per milliliter. Thus, 2 × 10⁻⁻⁻ cells can be suspended in 100 milliliter and processed through the STX in less than 30 minutes. Our VLX instrument can process 1 liter of concentrated cells or up to 2 × 10⁻11 cells in a single transfection run. I also want to point out that all transfections take place in sterile, single-use chambers called processing assemblies. We have different sizes of processing assemblies to accommodate different numbers of cells. Everything that comes in contact with a cell is single-use and disposable, so there is nothing to clean or sterilize in between uses. The MaxCyte technology is very simple to use. Users do not make any adjustments to the electrical parameters. They simply select a protocol optimized for their cell type from a drop-down menu. This is our current standard protocol list, which contains optimized protocols for over 80 cell types. If your cell of interest is not on this list, we also provide 10 optimization protocols ranked from low to high energy that should allow you to easily find conditions that provide good transfection efficiency and viability with almost any cell type. Now it is my pleasure to introduce Dr. Vanya Haberle and Dr. Cosmos from the IMP in Vienna in Austria, from Dr. Alex Stark's lab, who will now present their work about how high-throughput transcriptional reporter assays can enable the study of transcriptional regulation on a genome-wide scale. We are looking forward to your talk.
Thanks, Per. That was a great introduction into the advantages of flow electroporation technology, and I think it will be a nice segue into our next talk. Before we get to Vanya's and Cosmos' presentation, I want to remind everyone once again to submit your questions for the Q&A session at the end of the talks. Simply type your question into the Q&A box on the left-hand side of your screen and hit submit. We'll try to get to as many questions as we can. All right, let's continue with the presentations. Vanya, Cosmos, the floor is yours.
Thank you for the kind introduction. As introduced, in this presentation we will talk about exciting new reporter assay technologies developed in our research group and their application in studying transcriptional regulation. Our research group is interested in a very central question in biology: how the same genomic sequence can give rise to diverse cell types that make up complex organisms such as human. We know that different cells express different sets of genes that determine their morphology and function, and we also know which portions of the genome encode the majority of protein-coding genes. However, the information on when, where, and how much genes will be expressed is contained within the non-coding part of the genome in sequences known as regulatory elements. There are two main types of regulatory elements as depicted here: core promoters, which are located at the beginning of the gene, where the transcriptional machinery assembles and initiates low level of transcription. Enhancers, in contrast, can be located distally from the gene. They bind cell type specific transcription factors and increase the transcription from their target promoters to physiological levels in a cell type specific manner. The importance of non-coding regulatory sequences for gene expression and then ultimately for cell function is demonstrated by this genomic distribution of disease-associated single nucleotide polymorphisms in humans. As you can see, majority of such SNPs are not actually located within the coding sequence, but are rather in non-coding parts of the genome, for example, such as introns. And these non-coding sequences likely harbor regulatory elements whose disruption leads to dysregulation of target gene transcription. And this leads to disease, of course. And one such example are individual point mutations in the limb enhancer sequence of the sonic hedgehog gene, which is an important developmental regulator. And these point mutations lead to malformation of limbs in humans that manifest as polydactyly, depicted here in the pictures. The importance of this particular enhancer in the development of the limb is even more striking in the mouse transgenic model, where the mice with enhancer sequence deletion develop completely without limbs. The fact that apart from the limbs, these mice look normally and develop normally argues for a very restricted and tissue-specific activity of this enhancer. Furthermore, it was shown that the information for this tissue-specific activity is completely contained within the sequence of the enhancer. For instance, taking this enhancer out of its endogenous genomic context, placing it in front of a reporter gene, and testing for reporter expression in vivo in mouse embryo, as shown on the right side, shows that this enhancer drives transcription exclusively in developing limbs. Such experiments demonstrate the power of reporter assays, which is actually twofold. On one hand, the reporter setup recapitulates all key aspects of transcriptional regulation that happens in vivo, such as binding of specific transcription factors and recruitment of cofactors, which ultimately leads to transcriptional activation. But at the same time, it also allows to study functional properties of regulatory elements in a very controlled reductionist setup, where we can now change one variable at a time. And this can be used to study all components involved in this process, from both enhancer and promoter sequences, but also proteins that actually mediate their communication. And with this in mind, you will now hear in more detail from my colleague Cosmos how we utilize the power of reporter assays to study transcriptional regulation.
Thank you, Vanya. So traditionally, enhancer activity in cells is measured by luciferase reporter assays, where the enhancer candidate is cloned upstream of the reporter gene, and the expression of this reporter gene quantitatively reports on the activity of the tested candidate, as shown on the left side here. However, this approach only allows low throughput, as each candidate has to be tested individually, as the candidate sequence is not directly coupled to its activity readout. So to substantially increase the throughput, we developed STARR-seq, a massively parallel reporter assay. The key feature of STARR-seq is that we change the position of the candidate from upstream of the reporter gene to the 3' UTR of the reporter gene, shown on the right side, such that an active enhancer would transcribe itself and be present in the resulting reporter mRNA, what we now can use as a readout. And this direct coupling of sequence or of sequence identity to its activity now allows high throughput multiplexing and screening of highly complex library with millions of different candidate sequences at the same time. And to generate such a library, so we first sonicate genomic DNA, size selected, in our case, to 500 to 750 base pairs. We ligate the standard Illumina adapters to them, and via those adapters, we clone all those fragments into our STARR-seq vector in batch. And this yields typically 100 million to 1 billion different clones in our library. And one big challenge of screening such a highly complex library is its delivery to a large number of cells. To screen a genome-wide library, we need to transfect 2 to 5 billion cells. To reduce hands-on time, cells should be electroporated at a very high density. And we also require a high electroporation efficiency and cell viability. And of course, the electroporation has to work robustly and reliably. We also want to use our technology to transfect a wide range of cells, including insect and mammalian cells. To multiplex our assay even more per cell, we want to transfect high amounts of library DNA. So all previous systems we used so far, or tested, failed in one or more of those points. The first system that fulfills all those requirements perfectly is the MaxCyte STX electroporation system, which is superior in efficiency, cell viability, reliability, and also hands-on time. So now that we have successfully transfected our library, we can go back to the technical part of STARR-seq. So from the transfected cells, we isolate the resulting reporter RNAs from a pool of total cellular RNA, each containing the sequence of an active enhancer. We reverse transcribe them, specifically amplify the enhancer sequence via the Illumina linkers, and subject them to deep sequencing. The resulting reads are mapped back to the reference genome, where they not only indicate the position of each enhancer, but their abundance also reflects the enhancer's activity. These data are typically visualized as peaks in the UCSC genome browser, as I will show you on the next slide. In our first study, we applied STARR-seq to two Drosophila cell lines. On the one hand, as two cells which are derived from embryos, and the STARR-seq data of them are shown in blue in the genome browser screenshot in the upper panel. And on the other hand, ovarian somatic cells, or OSCs, which are derived from adult ovaries, shown here in red. We found thousands of enhancers genome-wide that were mostly non-overlapping between the cell types, and therefore cell type specific, as you can appreciate from the screenshot shown here, where each peak indicates the position of an enhancer in the genome, and the peak height reflects the strength of this enhancer. We also tested several of these peaks or enhancers individually in luciferase assays, and found a high agreement of STARR-seq peak height and luciferase signals. So together, this establishes STARR-seq as a genome-wide quantitative enhancer activity assay. Ovarian somatic cells are derived from the somatic tissue of adult ovaries, and therefore might have a counterpart in the living fly. So we produced 13 transgenic flies expressing GFP under the control of an OSC enhancer. 11 of the 13 enhancers drove GFP expression exclusively in the somatic tissue of the ovary, which confirms that STARR-seq enhancers can be functional in vivo in the right tissue, as exemplified here in the image on the right side, where GFP is only expressed in the somatic cells of the germarium that surround the germline cells, but not in the germline cells itself. We also use STARR-seq to study hormone or signaling responsive enhancers. In the upper panel, you see the experimental design. We transfected our library to S2 cells, treated half of the cells with ecdyson, and performed STARR-seq on treated and untreated cells. This allowed us to identify around 800 enhancers that become only active upon ecdyson treatment, as well as 900 enhancers that get repressed, which is shown on the upper right side. And to give you an impression how the data look like, we show a screenshot of the ecdyson-induced protein 75 locus with repressed enhancers that are shaded in blue and induced enhancers shaded in red. Recently, we could also identify enhancers that respond to Wnt signaling, which we don't show here, but if you're interested, we provide the reference at the bottom of the slide. So together, this demonstrates that STARR-seq allows to study enhancers under signaling-induced conditions and to identify hormone or signaling-responsive enhancers on a genome-wide scale. In all previous studies, we noticed a lack of STARR-seq enhancers around ribosomal protein genes, which led us to speculate if this is due to the use of a developmental core promoter for our screens, shown here on the right side. Therefore, we performed STARR-seq also with a housekeeping core promoter taken from a ribosomal protein gene. And I just want to mention here that both libraries we used are identical except the core promoter used. So performing STARR-seq with this housekeeping core promoter identified a completely different set of enhancers in the same cell type, as you can see from the screenshot, suggesting that housekeeping core promoters are activated by different enhancers that are incompatible with the developmental core promoter. Developmental enhancers are very cell type specific, as you see on the lower tracks and also seen before. Housekeeping enhancers are shared across cell types, as shown in the upper track in blue, and also the Venn diagram on the right side. And to investigate if this observation holds more general, we extended our analysis to 24 different core promoters of both classes, which confirmed that the ubiquitous activity of housekeeping enhancers, as well as the cell type specificity of developmental enhancers. And this led us to conclude the following model. In Drosophila cells exist two distinct major transcriptional programs, one for housekeeping and one for developmental gene regulation, which are separated by the specificity of their enhancers and core promoters. We could not only identify these programs, but also the underlying sequence features of their enhancers and core promoters. This also suggests that core promoters are not a neutral sequence hub where polymerase II assembles and transcription initiates, but that core promoters have a major regulatory function. Therefore, we also started to focus our research on core promoters. And so I will hand over now to Vanya again, who will tell you about the research on this.
Motivated by the observed specificity of enhancers toward different core promoters, as Cosmos just explained you, we decided to study core promoters in more detail, as there are still many open questions. For instance, we still don't know which sequences in the genome can actually function as core promoters, and what is the range of their strength. We also don't know whether different core promoters show different responses to certain types of enhancers. To screen for core promoter activity genome-wide, or in other words, for the ability of any sequence to initiate transcription when provided with an enhancer, we modified our original reporter setup. As in STARR-seq protocol, we tonicate the genomic DNA, but now into shorter fragments, ranging from 150 to 200 base pairs, which is closer to core promoter definition, and we clone them in batch into a reporter plasmid. However, this time we put the fragments in place of a core promoter, which is just downstream of a fixed strong enhancer and upstream of an open reading frame. We again need to deliver this highly complex plasmid library into large number of fly S2 cells, which we do with high efficiency using MaxCyte STX electroporation system, as noted before by Cosmos. In the cell, the sequences that can respond to given enhancer and initiate transcription will give rise to reported transcripts. And their abundance will directly reflect the core promoter activity of each initiating fragment shown here in different colors. We then ligate the RNA linker to transcripts, which contains a unique molecular identifier that allows us to count original individual transcripts even after amplification and sequencing. This is followed by reverse transcription and amplification of reporter fragments, which are then subject to high throughput sequencing and finally mapped back to the referent genome. And map tags, shown here in blue at the top of the slide, indicate the exact transcription initiation position and quantify the abundance of initiation events genome-wide. Since we want to measure responsiveness of genomic fragments to a given enhancer, we have to account for the possible intrinsic activity of the candidate fragments themselves. So we repeat the screen with the same fragments using a reporter that contains no enhancer and normalize the tag counts from the previous screen to this one. This gives us a measure of responsiveness of each genomic fragment to that specific enhancer. The genome-wide results that we obtained showed that only specific discrete genomic fragments can act as core promoters and initiate transcription, whereas the rest of the genome has negligible activity. As exemplified by this genomic locus, the signal coincides with annotated gene starts, and the transcription is initiated in a strand-specific manner that follows the direction of the gene. And what this means is that sequences of endogenous core promoters are clearly distinct from the rest of the genome, and that they encode the ability to respond to enhancers, as well as directionality of initiating transcription. Visualizing the data genome-wide and comparing two biological replicates demonstrates, first of all, high reproducibility of our screen, but more importantly, it also reveals very wide range of core promoter responsiveness to a given enhancer, which goes up to 1,000-fold difference. Main implication of this observation is that the actual transcriptional rate is a combination of enhancer strength and core promoter responsiveness to that enhancer. And interestingly, we found that core promoters with different responsiveness are used in the genome for different purposes. For instance, the most responsive promoters in the top right corner are usually coupled to genes encoding cell type specific transcription factors, which need to be turned on quickly and have to be expressed at high levels. In contrast, genes encoding for cell type specific metabolic enzymes usually have less responsive promoters, as they do not have to be so tightly temporally regulated. The knowledge we gained from our screen can also be used to optimize gene expression constructs. One could, for instance, couple a very responsive promoter to a very strong enhancer to boost the transgene expression. And with this, I would like to conclude this part and remind you that all the results presented so far were done in fly cells. So my colleague Cosmos will now briefly present an interesting application of described technologies in studying biology of human cells.
The project I'm presenting now was a collaboration with Johannes Zuber's lab here at the IMP in Vienna. So they found that the cofactor BRD4 drives MYC expression in leukemic cells, and that inhibition of BRD4 with the small molecule inhibitor JQ1 resulted in shutdown of MYC expression, which eventually killed those cells. However, some leukemic cells are resistant to JQ1 by re-establishing MYC expression around 24 hours after JQ1 treatment, shown here on the right side. To understand why these cells started again to express MYC, they profiled the H3K27 acetylation chromatin mark in the MYC locus of sensitive and resistant cells, and found substantial differences in the chromatin landscape downstream of MYC in the neighboring gene PVT1, as shown by this genome browser track on the bottom. As K27 acetylation is believed to coincide with active enhancers, we hypothesized that changes in the regulatory landscape of MYC might confer this resistance. So we decided to profile the enhancer landscape of the MYC locus in a resistant cell line using STARR-seq. For that, we cloned a focused STARR-seq library from bug DNA covering 3 megabases around MYC. We transformed the resistant cell line K562 with this library, treated half of the cells with JQ1, and performed STARR-seq on treated and untreated cells. This allowed us to identify an enhancer in the first intron of PVT1, which becomes strongly activated upon JQ1 treatment, here shown with the red arrowhead. Actually, it's the strongest activated enhancer in this entire experiment. And this also coincides with differences in the chromatin landscape, as shown in the genome browser tracks above. So the model for the resistance to BRD4 inhibition is the following. JQ1 treatment inhibits BRD4, which leads to the shutdown of MYC expression. At the same time, however, Wnt signaling is activated in resistant cells, which leads to an activation of a Wnt-dependent MYC enhancer. And through this enhancer, the inhibition of MYC by JQ1 is bypassed and MYC expression is re-established, what leads to the observed resistance. So with this example, how STARR-seq can help to understand mechanisms related to human disease, we are at the end of this webinar, and I would like to summarize what we've told you so far. Today, we showed you that STARR-seq is a quantitative enhancer activity assay that allows to draw genome-wide enhancer activity maps. We also showed that STARR-seq allows to study signaling-induced enhancers and to identify them on a genome-wide scale. STARR-seq also identified two major transcriptional programs in Drosophila that are separated by the specificity of their enhancers and core promoters. Further, we showed you how STARR-seq can help to identify disease-related enhancers in human cells. We also introduced STAP-seq, a method that allows to study the responsiveness of core promoters towards enhancers, which revealed that core promoters are important gene regulatory elements rather than neutral assembly sites for RNA polymerase II. And finally, we demonstrated that the MaxCyte STX electroporation system is perfectly suitable for the delivery of highly complex libraries and performs outstanding in transfection efficiency and cell viability. So taken together, the MaxCyte STX enables genome-wide functional assays to study transcriptional regulation in a great variety of cells. We want to thank our boss, Alex Stark, and the entire Stark Lab, as everyone in the lab was involved in one or more of the projects we presented today. We also want to thank our great service facilities and the next generation sequencing unit here at the campus in Vienna for the great support. We also want to thank MaxCyte, especially Pierre and Eric, who helped to set up the electroporation system here, and Jason, who helped to set up this webinar. We also want to thank Jen for the great support in conducting this webinar. And of course, we want to thank our funding sources. And finally, we want to thank you for your attention.
Thanks a lot for this interesting presentation. I think we all enjoyed it. Now, in conclusion, I'd like to highlight the key benefits of MaxCyte's scalable cell delivery platform. With a MaxCyte system, you can work with the appropriate cell type. Users can produce a wide range of molecules. High productivity stems from high transfection efficiency and viability. MaxCyte's electroporation-based platform is much more consistent and scalable than chemical-based transfection methods. Efficiency and scalability enable high transient productivity as well as more efficient production of stable pools and clones. Higher productivity, in turn, allows customers to dramatically shorten development timelines. And with this, I would like to end and thank the audience. And yeah, now we are happy to take questions.
Thanks, everyone. Vanya Cosmos, excellent presentation. I think you gave the audience some great information about the benefits of a reporter assay and showed them how useful it can be for their own research endeavors. So we thank you for that. Before we start the Q&A session, I want to remind everyone once again, this will be your final chance for submitting questions for our speakers. So hurry up and send them in now. We've already got a few questions, great questions that have rolled in, so send them in and we'll try to get to as many questions as we can. All right, so let's begin the Q&A. Give us a few moments on our side to get all the particulars straightened out, and we'll begin the Q&A presently. Guys, there, maybe. No? All right. So, Per, are you there? Maybe we'll switch to Per for a question.
Yes, I'm here.
Per? Okay, Per. So we'll switch to you. Maybe Cosmos and Vanya have a little technical difficulty for a second. So, Per, one of the audience members would like to know: is it possible to freeze the cells post electroporation?
Yes, absolutely. So there's no impact on the performance of the cells if you freeze them direct after the transfection. And even if you analyze or do the analysis weeks or months or years after it, there's no impact.
All righty. Thank you very much. Cosmos, Vanya, are you guys back on?
We are on.
Okay. So we'll go back to our first question we have. So like I said, one of our audience members would like to know if it's possible to perform a genome-wide screen in human cells using STARR.
Yes, definitely. So with the current protocol, or the protocol we released so far in our previous papers, we only recommended to do screens in human cells in like focused or restricted libraries because of the huge effort you have to take to do it genome-wide. But we will, like in the next weeks, release a new optimized protocol for genome-wide screens in human cells, which will be pretty exciting. Yeah, and this is partially also thanks to MaxCyte technology, as you need to really transfect a huge amount of cells with this high efficiency. But you will see this. We put this on bioRxiv, I think, in 2 to 3 weeks.
All righty, thank you very much. We have a couple more questions for both of you guys. One of our audience members would like to know, what happens if you use different inhand or refseq?
Yes, so what we presented in today's presentations were results that we obtained from using the one-fixed enhancers, which is a very strong developmental enhancer that we actually identified in STARR-seq. But we also performed experiments with a different enhancer, which was also very strong developmental enhancers, and what we saw was actually that the results were pretty similar. So no matter which developmental enhancer we use, we identify the same promoters and they have the same strength or same responsiveness. And also one interesting aspect is that we performed STAP-seq with the same enhancer and with a different enhancer in different cell types. And when we move to another cell type, we also see very similar results, which suggests that promoter responsiveness is also independent of the cell types in which we do the screen.
All right, thank you very much, Vanya. Both, again, for you guys, here's another question. How did you decide to use a fragment size of 500 to 750 base pairs in your STARR-seq library generation?
Yeah, so this is basically a combination of what is known of Drosophila enhancers and also try and error, where we basically in luciferase assays basically tested fragment sizes of known enhancer, how small or big we can go, and we basically found that this 500 to 750 base pair works best in our screens. And a similar thing, so for humans, for example, we now recommend to use 1 to 1.5 kb fragment size, because we found that the smaller fragments are giving not that nice signals as if you take a little bit bigger fragments. Yes, so this is a try and error plus the plus literature-based research, what we did on that.
All righty, thank you, Cosmos. Another question for you guys. An audience member would like to know about the supercore promoter will have a basal level of expression. How high is the background level of expression? And can you use this technique to identify inhibitory regulatory sequences as well?
Yes, so there I can say, yes, the supercore promoter has some basal levels of expression like almost every core promoter has, which if you look at our screenshots or the tracks, you basically see very low levels of STARR-seq transcripts, all around the genome. And then only like where like an enhancer sequence will give rise to much more transcripts, which then build up those peaks you see. And so we regarding to screen for inhibitory regulatory sequences, we did some pilots on that. So it certainly doesn't work if you only look at the basal level of the supercore promoter. So what you need to do is you need to put a strong enhancer in front of the STARR-seq cassette to basically drive it constitutively, and then you can identify repressors. We found we found repressors doing this, but we also found regions which basically destabilize the transcripts and are not really repressive. Yes.
All righty. Thank you. We have another interesting question for you guys. Could you use STARR-seq to identify or pH-sensitive enhancers?
So here we can also say certainly yes. As we showed like in the presentation, you can basically use STARR-seq to identify enhancers for perturbations or for signaling pathways and hormones. And we also, that's an ongoing project in our lab, we basically use or perform STARR-seq under heat shock conditions, which also gives really nice results. We didn't we didn't look into pH-sensitive enhancers yet, but as all perturbation worked so far to identify like corresponding enhancers, I'm pretty sure this is also possible.
All righty, thank you. And one last question. How do you normalize for the level of plasmid DNA in the cells, i.e. the more plasmid DNA, the more transcripts, which is nothing to do with.
Okay, so what we normally do is we, of course, sequence our input library to see if all fragments are equally represented. And we usually have a very nice even coverage across our input DNA. And since each input fragment has the same chance of being electroporated or to end up in the cell, we think that this is normally enough. So we normalize to this input coverage that we sequence. But we also checked using DNA, plasmid DNA that we extracted back from the cells to actually account for this difference in electroporation that can happen. And we also see pretty even coverage of our input library that ended up in the cell.
All right, thank you, Vanya. Actually, we have a couple more questions that just came in for you guys. Are there biomedical applications of STARR-seq and STAP-seq?
Yes, I would say definitely. I mean, what we showed at the end of the talk, we were able to identify enhancers which are responsible for drug resistance, but you certainly can also use STARR-seq to basically to study or screen for underlying enhancers for either drugs which are on the market already or for testing new drugs, I would say.
All right. And another question from our audience member would like to know, how long does STARR-seq and or STAP-seq take to perform with new cell line from start to finish?
So I think the first part is that you need to basically get the electroporation of this cell line right, and with like if you use MaxCyte, for example, I mean they usually provide you with like nice optimized protocols. So if you basically so from transfection until you get your sample sequencing ready, that's only a couple of days. You might also need to clone the library, which is another couple of days. So I would say if you start from very beginning to the very end, you can do this like until you get it onto the sequencing machine like in 2 weeks if you work hard on it.
All righty. Thank you, Cosmos. And I think this would be the last question for today. Can you find strong enhancers and core promoters using STARR-seq and STAP-seq?
Yes, of course. I mean, this is what we do because, I mean, both methods are quantitative. So, meaning like strong peaks in STARR-seq usually indicates strong enhancers, and also in STAP-seq as we count individual molecules. We of course know the strength of the core promoter, and so having both knowledge basically together, you can even basically boost your transcription by combining like a strong enhancer with an even more strong core promoter, or you can also use it to fine-tune your expression system by using a strong enhancer with a medium strong core promoter, or medium enhancer, medium core promoter. So it basically like gives you the toolbox to build like tailored expression systems.
Alrighty, thank you, Cosmos. And with that, we've come to the end of our webinar. So I want to remind everyone that the webinar will be archived on our site at www.genengnews. It'll be archived for up to a year. So if you missed parts of it, you can watch it again, or you can feel free to forward the link to your friends and colleagues, which we always recommend. I'd like to thank Pierre, Vanya, and Cosmos again for their informative presentations. And I'd like to thank the audience for their attention and very thoughtful questions. And a very special thanks to MaxCyte for sponsoring this webinar. So hopefully we'll see you again at another Gen webinar in the near future. Goodbye for now.